this is Dr. Neil Cross, and I'll be teaching you about the genetics of cancer in this session, and specifically I'll be teaching you lots of genetics concepts that relate to cancer. Firstly, about why cancer is a disease with a genetic basis, and sometimes that can mean it is a genetic disease as a classic inherited disease, but it also is a disease, even where it's sporadic, it is driven by acquired somatic mutations. And without genetic mutations, cancer will not develop. We're also going to look at the multi-step and monoclonal nature of tumour formation, which basically means that cancers arise from a single cell and then acquire lots of multiple mutations in key pathways that drive the cancer formation. And to help explain these concepts, I'll be covering a whole series of definitions and concepts. Anything that's bold and underlined is either a definition or a concept that you need to understand. Some of these are genetic phenomenon, such as microsatellite instability, Knudson's 2-hit hypothesis. All of these things I will go over, but they'll be highlighted in the lecture slides. So firstly, some basic concepts about what is cancer. Cancer cells are derived from what were previously normal cells within the body. So cancers arise from normal cells that have acquired genetic mutations, and then those genetic mutations prevent the cell from regulating its own normal growth and cell death decisions. What we find that cancers from the same site, so cancers from the same site such as breast cancer, lots of breast cancers often behave in a very similar way. They have often the same genes affected. Similar with colon cancer, the same genes tend to be affected in most colon cancers. And this allows us to understand what is going on, what is driving the development of those cancers, but it also gives us clues as to which genes are normally regulating and preventing aberrant growth in individual tissues. So the mutations that occur in cancer are permanent genetic mutations. So we will have mutations in things like growth factor receptors that will drive aberrant and excessive growth. All of the daughter cells will have the same mutations as the initial cell, and then as time goes on, different clones of cells acquire different mutations, and the tumour can change its genotype and phenotype, sometimes subtly, sometimes significantly, but these are all permanent genetic mutations. So cancer is a disease with a genetic basis, and these changes to the genome of the cancer we call somatic mutations. So somatic mutations are mutations that have acquired that are different to the constitutional DNA of the person from which the cancer came from. And in the previous lecture I talked about genetic sequencing, and what we often do is look at a patient's normal DNA taken from blood lymphocytes and then compare that to the tumour DNA, and anything that differs between the two is a tumour-specific mutation. The sorts of mutations that cancers have are all of the types of mutations you'll have come across, so point mutations, either changing one amino acid to another, or changing one codon to another, and that can be a different amino acid, or it could be a stop codon. We look at things like gene amplifications, where an entire gene can be copied and copied again, so you maybe have dozens of copies of the same gene tandemly duplicated back to back, and that results in increased gene expression. This is associated with oncogenes. Point mutations can be associated with tumour suppressor genes if they're inactivating, or oncogenes if they are activating, and I'll come on to those down here. We also see things like chromosome instability, where the chromosomes themselves just seem to break up and recombine in weird configurations. We'll see deletions of entire chromosomes or small regions of chromosomes, and we call this loss of heterozygosity, partly based on how we localise where the deletions are, and I'll define loss of heterozygosity. Some cancers, particularly colon cancers, have something called microsatellite instability, and this is where the cancers are unable to repair their own DNA due to a DNA mismatch repair defect, and then that causes microsatellites, which are things like CA repeats, from expanding and contracting. So that's a feature of those particular cancers. We can also have epigenetic gene silencing, and that tends to affect tumour suppressor genes. So the two main genes that we're interested in here, the two families, are the tumour suppressors. These are growth inhibitory and pro-apoptotic genes. or their protein products are. So it's usually one or the other, sometimes both. So these become inactivated by a somatic mutation, but also in some cases we can have germline mutations of tumour suppressor genes, and they can give rise to hereditary cancer syndromes. There's a separate bunch of tumour suppressor genes which are DNA repair coding genes, things like the mismatch repair system, which I'll talk about later. So those are all tumour suppressor genes, and those mutations are what we call loss of function, because we've got 2 copies of almost every tumour suppressor gene, and loss of 1 copy of that gene doesn't really have much of an effect, but loss of 2 copies does have an effect, and this will be really important when we come on to something called Knudson's 2-hit hypothesis when we look at hereditary cancers fairly shortly. In contrast, we've got oncogenes. Oncogenes are growth stimulating genes. So proto-oncogenes are our normal, things like normal growth factor receptors and downstream signaling molecule proteins. So proto-oncogenes become mutated into oncogenes, and they're typically activated into a constitutively active form by somatic mutation. So that can either be overexpression or mutation of a key amino acid, of a key codon, changing a key amino acid to something else, and that renders the oncogene coded protein, the oncoprotein, into a constitutively active form. So we'll look at some key tumour suppressor genes and oncogenes later, but I'm going to introduce to you the multi-step nature of cancer, and this is basically what we call the multi-hit model of cancer. And the multi-hit model of cancer explains how tumours develop from initially a normal cell. So down here we've got the colon cancer model. It was devised back in about 1990, also known as the Vogelstein model, because it was devised by someone called Vogelstein. So we start off with a normal epithelium. We tend to lose a tumour suppressor gene called APC, adenomatosis polyposis coli. That's part of the Wnt signaling pathway. For anyone who's studied cell biology, you'll know what that is. Everyone else might not matter. The key thing about APC, there are 2 copies of it, because it's on one of our autosomes, and you lose 1 copy first, and then that same cell loses a second copy, so you end up with a cell which is APC null. Then we get some other things happening, epigenetics, silencing of some tumour suppressor genes. Then the next thing that happens that's a really important gene is activation of the KRAS oncogene, and this is downstream of growth factor receptors, and this drives unregulated proliferation. This APC gene alters differentiation, so when cells have got intact APC, they tend to proliferate for a short period of time and then differentiate into normal colon cells. If they lose APC, they just continue proliferating. KRAS synergizes with this to drive more rapid proliferation. We get some other things happening, but particularly here we get loss of the p53 gene, and the p53 gene regulates both growth and DNA repair and programmed cell death. So this is really important. Now all of this is happening before we've got a fully fledged cancer. Loss of p53 seems to be the final step from being an adenoma, which is a benign tumour, into a carcinoma, which is a cancer which is locally invasive within the tissues by definition. Now we know that cancers arise from a single cell, and that is we know this from methylation studies in females who develop cancers. So if you think back to X-inactivation, which you must have covered at some point in a previous life, early in embryogenesis, X chromosomes in females, there are 2 X chromosomes and both of them are active. Early in embryogenesis, these are randomly inactivated, so one cell expresses the maternal X chromosome, one cell expresses the paternal X chromosome. So you can have patches of cells that are expressing the maternal X chromosome, others are expressing the paternal X chromosome. In tumorigenesis, what we find is in tumours from females, the tumours, all of the cells within the tumour have the same X chromosome which is active, which proves beyond doubt that cancers arise from a single cell. Otherwise, if cancers arose from multiple cells, you would expect different, a mixture of X chromosomes being active within the tumour. So this is proof that tumours are monoclonal in origin. There is the odd exception to this, and that's where patients develop multiple independent cancers. Now, males develop prostate cancer quite frequently, and it's not unusual for a man to have two separate prostate cancers, so they would be what we call polyclonal. Also, some women develop multiple breast cancers, sometimes in the same breast, and when they sometimes develop truly as two independent cancers, and in that situation one cancer might have the maternal X chromosome active and one might by chance have the paternal X chromosome, but they are two independent cancers. Within each cancer the same X chromosome is activated or inactivating, proving that all of those cells within that cancer came from a single cell. Okay, this slide just shows how tumours can become very heterogeneous even from starting from a single cell. So this cell is the original cell that has acquired mutations, and then subsequent daughter cells have picked up other mutations. And as you follow this line, this is the sequence of events that's happened in this particular cancer. It's very much like a Darwinian evolution. And lots of cancer biologists talk about Darwinian evolution because as a cancer develops a mutation here, so loss of 17p, that's loss of the p53 gene because that's where it resides, that will give this clone of cells a selective advantage over the rest of the cells, and it will proliferate and refuse to die. And then that might then pick up another mutation such as CDKN2A loss, which will allow the cell to proliferate faster. And then there'll be subsequent mutations like this loss of chromosome 13 is loss of the retinoblastoma gene, that's where it lives. And then subsequently you get other changes. So each change is giving that clone of cells a selective advantage. And this can actually give rise to quite heterogeneous populations of cells within a tumour. They all stem from the same original starting cell, but they sort of become cancerous by complementary or similar pathways. And it's very much like clonal evolution going on within an individual tumour. So this slide shows a more up-to-date Vogelstein model of colon cancer development, and it shows both tumour suppressor genes in red and oncogenes here in green for the initiating step. Now in colon cancer we can lose the APC gene. That's often the first hit. So a cell loses the APC gene, the second hit is usually the loss of the second copy of that gene, a second somatic mutation. That will create what we call an aberrant crypt, and that will then progress onwards. However, we can also have that first hit being an inherited mutation. So the APC gene is adenomatosis polyposis coli. If you inherit a defective copy of that, that causes a disease called FAP, familial adenomatosis polyposis, and this is where you get lots of colon polyps throughout your colon. So the first hit can be an inherited mutation, and then the second hit can be a somatic mutation. So in both situations here, whether it's a sporadic cancer, you get this first and then this, or a hereditary cancer, you get this mutation inherited and then you get this. They both lead to the same point. You can also get to that same point due to a single mutation in beta-catenin. Beta-catenin is linked to APC, and a dominant oncogene mutation of beta-catenin can have exactly the same effect. So that is what we call the first initiating step of cancer. What we often then get is some sort of genomic instability, so enhanced mutation rates, sometimes some epigenetic changes. Then we've got KRAS, which I mentioned on the previous slide, and then our other changes. So these initial insults can be summarised by this slide. So the first hit, it comes from either bad luck, and we know that the majority of cancer is down to bad luck. It can also come down to a high carcinogen burden, so a diet that's low in vegetables and high in burnt meat. You will be taking in more carcinogens and you're more likely to develop colon cancer. So that's your first hit. The first hit could also be hereditary predisposition, as I've just mentioned. So that's your first hit. You've lost one copy of your APC gene. Second hit is also, bad luck, high carcinogen burden. Now, as we'll see with hereditary predisposition, it sort of doesn't matter what the carcinogen burden is. This second hit is always going to happen. And it's always going to happen because every single cell in a patient's body is deficient for one copy of APC, and it only takes a carcinogen in any one cell to inactivate that second copy of APC. So this is what happens in hereditary cancers. You inherit one defective copy, and then every single cell in your body is at risk of losing a second copy due to a random carcinogenic event. We then get subsequent genetic effects, genetic mutations which enhance genomic instability. So we sometimes get something called a mutator phenotype, where cells can't repair their own DNA, and then this will drive the subsequent accumulation of additional genetic damage, leading to all of these additional steps. Now, if you didn't have this mutator phenotype, these mutations here wouldn't happen by chance. The chances of that happen is really, really small. But if your mutation rate is 100 times higher than a normal cell, then those mutations can readily occur. So I've alluded to hereditary cancers on the previous slide, and this slide summarises some of the main genes which are inherited that cause hereditary cancer syndromes. So in these situations, you inherit one defective copy of the gene that is defective in every cell in that patient's body, and then any one cell can then have inactivation of the second copy, and that is the cell that is going to go on and develop into a tumour. So RB1 codes for the retinoblastoma protein. This controls cell cycle arrest, and patients who inherit an RB1 mutation develop a retinoblastoma, either unilateral or bilaterally in the eye. APC, as we've heard, this is the Wnt pathway. It controls growth and differentiation. If you inherit a defective copy of that, you will develop multiple independent tumours in the colon. P53 controls DNA repair, cell cycle arrest, apoptosis, and if you inherit a defective copy of that, you will develop Li-Fraumeni syndrome and likely develop multiple primary cancers in multiple different sites. And then we've got BRCA1 and BRCA2. Those are inherited and often give rise to bilateral breast cancer. HMLH1 and HMSH2 are DNA mismatch repair enzymes, and they result in multiple colon tumours. Now, the interesting thing about these is they're all dominantly inherited. There are a few cancers which are recessively inherited. They tend to be more sort of metabolic disorders, but most are inherited dominantly, yet all of the tumour suppressor genes are recessive in activity. And you think about recessive genetic diseases, you need to lose both copies of the gene to have an effect. Well, in cancers, it's exactly the same. You inherit one defective copy. Now, because every single cell in the body is defective for that, the chances of any one cell acquiring that second mutation to have a phenotypic effect is near enough 100%. So therefore, if you inherit one defective copy of that gene, then you are pretty much certain to develop the disease, the hereditary cancer syndrome, and therefore these are inherited in a dominant, autosomal dominant manner. And this is illustrated here, and this phenomenon is called Knudson's two-hit hypothesis. So it's really worth clicking on this link here in the PowerPoint version of the slides and reading about Knudson. Alfred Knudson, he figured out this two-hit model of tumour suppressor gene inactivation before tumour suppressor genes had been cloned and identified, but he calculated it mathematically by studying retinoblastoma. So I'll take you through these two different scenarios. This is a non-hereditary, or shall we say sporadic, retinoblastoma. At conception, everything is normal. So with respect to retinoblastoma, at conception, everything here is normal. In one cell, by poor random chance, one copy of the retinoblastoma gene has been inactivated. Okay, so all daughter cells from that cell will be RB1 deficient. It then takes a stroke of very bad luck for one of those daughter cells to then subsequently pick up a second mutation in the retinoblastoma gene on the other chromosome 13 homologue. So we then end up with a cell which is completely deficient in retinoblastoma protein, also known as PRB, and that PRB protein regulates the cell cycle. So the fact that we've got no PRB protein because the RB1 gene is knocked out, both copies, we end up with this tumour, and this develops quite rarely, about 1 in 30,000 live births, and these occur in a single eye. One single tumour in each patient that develops it. It's very rare, but you only get one tumour. That's a really important point. In contrast, this side shows hereditary retinoblastoma. This patient here at conception has inherited one defective copy of RB1. All of the daughter cells, labelled A, are defective for one copy of RB1. It only takes one of these cells, of which there are millions in the developing retina and optic nerve, to lose the other functional copy. So at point B, here, we've got to the point where we have got a double deletion or double knockout of RB1, and then that will go on at point C to form a tumour. Now, the tumour formation is very common in the RB1 carrier population, and effectively the overwhelming majority of people who inherit one of these mutations develops the tumour. So effectively it's dominantly inherited, whereas over here the patient was normal for RB1 at conception and develops a single sporadic tumour. Over here, at conception, this patient was destined to develop at least one cell becoming double knockout for RB1, and in fact these patients often develop multiple tumours and can have tumours in both eyes. And this is a classic feature of retinoblastoma of bilateral presentation. And bilateral presentation also occurs in other hereditary cancer types as well, particularly breast cancer. So as shown here, sporadic cancers arise due to disruption of both copies of a tumour suppressor gene. So in breast cancer, those cancers often lose BRCA1. In retinoblastoma, we lose the RB1 gene. That is lost in both hereditary and sporadic retinoblastomas. It's a rare event to occur sporadically. It's a common event to occur in the hereditary cases. So this is why it's called the two-hit hypothesis, proposed by Knudsen in 1971. And in effect, what Knudsen observed is that patients who present very early with a retinoblastoma often then presented with the second tumour in the other eye a few months or a couple of years later. And he figured out that this could be due to an inherited predisposition, and it could be down to loss of two copies of the same gene that occurs, and you develop one tumour earlier and then another one just happens to occur a little bit later. What he also realised is that patients who develop only one tumour, they tended to present later. So he figured what was going on there is they develop later because the patients are born normal for that particular gene. They lose one copy by bad chance and then lose a second copy by poor random chance, and therefore those tumours develop later because two hits are required, whereas for the hereditary cases only one hit is required, the other hit coming from the inherited mutation. So this slide summarises what the disease presentation is for sporadic versus hereditary cancers. For sporadic cancers, you tend to get a unilateral single tumour. So that could be a single breast tumour, a single eye tumour. In the colon, it would just be a single tumour developing in the colon. You tend to get later presentation. So with retinoblastoma, it's maybe 3-4, sometimes 3-4 years, whereas with hereditary forms, it's earlier. Sometimes these are all already present at birth. For sporadic tumours, They can be common, they can be rare. Breast cancers are common. We know 1 in 9 will develop breast cancer. Retinoblastoma, 1 in 30,000, a very rare tumour. However, within the hereditary population, so if you inherit a defective copy of RB1, your chance of cancer is close to 100%, or it might be around 50% for BRCA2. So it's dominantly inherited, but it can have what we call a variable penetrance. So you can inherit a mutation of BRCA2, and it's dominantly inherited, but you don't actually always get cancer. About half of cases develop cancer. We'll call that variable penetrance. But the key point to remember, sporadic cancers are unilateral, hereditary cancers tend to be bilateral or multiple tumours. Sporadic cancers are later presentation, hereditary cancers are earlier presentation, and the hereditary forms tend to be inherited in an autosomal dominant manner, but sometimes with less than 100% penetrance. Okay, so I'm going to pause now. This is the end of the section on hereditary cancers, and I'm now going to, the next video is going to be more about the types of genetic mutation that occur in cancers. Okay, so have a look out for that next video.